Welcome back, ladies and gentlemen. So today we are going to get into the actual processes of how we take that message in your DNA molecule and actually create the proteins that those instructions in your DNA actually code for. So remember when we're talking about this flow of information in our cells, everything flows in a single direction. It flows from that DNA in your nucleus into these RNA molecules that we create from that code. And that code then goes out into your cytoplasm and actually builds the proteins, the amino acid chains itself, this polypeptide. So the two major steps that occur in order to get that information from your nucleus to the final product is in the process of transcription and translation. So remember that transcription is converting the message or the set of instructions in your DNA, and we're making a copy of it. We're creating that messenger RNA from that. So if you think back to considering DNA as this giant cookbook for every cookie recipe, for example, that could exist in the world, all of those recipes are stored in that big cookbook in that DNA in the nucleus. And that cookbook never leaves. But your factory floor, the actual production of those cookies, for example, has to happen outside that nucleus. In this case, in your cells, it's going to occur out in your cytoplasm. So the whole point of transcription is to copy just the message that I need. If we're using the cookie analogy, just the recipe for, say, chocolate chip cookies. And we're going to copy that chocolate chip cookie recipe into that messenger RNA molecule. That messenger RNA then can leave the nucleus through pores in the nuclear membrane and go out to your cytoplasm, go to the ribosomes, which is where we do the translation, where I'm going to take that DNA RNA language, that nucleic acid language, and literally translate it, change it from one language to another. So when we look at transcription, to transcribe literally means to copy something. There was an entire profession of scribes where all those scribes did was to sit there and copy information, either from a book or from somebody who was verbalizing that information, and that scribe was writing it down for them. That's the idea, and that's where that word transcription comes from, copying a message. Translation, if any of you are taking German or Spanish, right, you translate text or you translate words all the time. You're taking it from English to Spanish or English to German or vice versa. You're changing the language that you're speaking. That's exactly what your cells are doing as well. They're taking this nucleic acid language in this messenger RNA code and translating it into amino acids or into protein language. So today we're going to focus on transcription and what happens and how this process actually occurs. So again, remember, transcription is to write or to script the code and move it then ultimately across the membrane. So if I see where these words trans and script come from, script means to write the code, and then trans means to go across or to move across something, in this case, the membrane of your nucleus. So transcription is all going to happen in the nucleus because I'm going to need the DNA code to make the copy into messenger RNA. And so DNA is housed in your nucleus, and so that's where this process is going to occur. We're also going to have an enzyme here that's going to be necessary to make this process happen, and that enzyme is called RNA polymerase. Now, we've already come across DNA polymerase, right? Polymerase, the polymer, right, is a long molecule of smaller pieces. When we talked DNA polymerase, right, that polymerase enzyme's job was to build all these little nucleotides, put them together to make DNA. Well, RNA polymerase is going to do a very similar job, but instead of DNA nucleotides, we're going to use RNA nucleotides. Something to keep in mind as we're making the copy of this code, that DNA molecule is going to give me the template, just like it did for DNA replication, to know what base to come and bring in next. But just remember that there's no T's in your RNA molecule. So when we match A, it's going to match with U in RNA instead of the A to T base pairing we've been talking about with DNA thus far. And you can see that down here, right? So T will still match with an A RNA, but A now goes with U in that messenger RNA molecule. Okay, So we're going to break transcription into a couple of steps, and we're going to start here with this idea of just initiation. To initiate something means to start it. So to start transcription or to initiate transcription, I need to get access to the DNA molecule itself and actually be able to read the code or read the set of instructions that are there within those nucleotide bases. So RNA polymerase is going to attach to your DNA molecule wherever that set of instructions, wherever that gene starts, and open up the DNA molecule. It's going to unwind it. So we're going to sort of relax the double helix, and we're also going to separate the two strands. Then elongation is the next step, where I take that RNA polymerase, and as it moves along that DNA molecule, we're base pairing based on the original DNA molecule, building that messenger RNA as we go. All right, remember that A is now going to pair with U in our RNA molecule, and C will continue to pair with G in RNA, just like it did back in DNA. And we'll continue to add these free RNA nucleotides that are just kind of floating around in your cells until we get to the end of the instructions, or the end of the gene in DNA. Once we hit the end of that gene, there are signals within your DNA molecule to say, boop, you're done. All right, and so we hit this termination step. And to terminate something literally means to stop it or to end it. So once we get that signal from DNA to say, yep, we're cool, we're done, we're at the end, that RNA polymerase reaches the end of the gene, all of the components come off, the RNA polymerase detaches, the messenger RNA detaches, and your DNA is able to go ahead and reform these hydrogen bonds, twist itself back into its double helix, and transcription has now come to an end. We now have a full messenger mRNA molecule that is now going to leave the nucleus to be able to then build the proteins out in our cytoplasm. So if you think of a gene structure, think of it like a capital letter at the beginning of a sentence. That capital letter is going to tell that RNA polymerase, hey, cool, this is where this information is going to start, and that's going to help initiate initiation by RNA polymerase binding to the DNA at what is essentially, in our analogy, the capital letter. Elongation is kind of like reading the sentence. What is the actual information that that sentence is telling you? Well, that's what's happening here. And so the elongation is essentially that RNA polymerase reading the sentence and making a copy of it. In this case, taking that DNA and copying it into messenger RNA. We get to the termination then, and well, what do you find at the end of a sentence? A punctuation mark, a period, a question mark, an exclamation point. That is essentially what's going on in your genes. Your DNA polymerase will continue to read that sentence or read that DNA code until it gets to the instructions to stop. And just like a sentence, it's very similar in the sense of hitting that punctuation mark, and now you've got the full message in that messenger